Hi, and welcome to 4.2 Rules of Components of Cell Membranes. Okay, cell membranes part 2. Okay, so we've talked about phospholipids and a little bit about proteins, different types of proteins and glycoproteins. And we have seen that cell membranes contain several different types of molecules, even more than what we've discussed, including cholesterol and glycolipids. So there are proteins and glycoproteins, which we discussed in our last, but I'll give you a small refresher. Okay, so phospholipids form the bilayer, which is the basic structure of the membrane. And because the tails of phospholipids are nonpolar, it is difficult for polar molecules or ions to pass through membranes. So they act as a barrier to most water-soluble substances. So, for example, water-soluble molecules like sugars, amino acids, and proteins cannot leak out of the cell, and unwanted water-soluble molecules cannot enter the cell.
Okay, so one thing we haven't talked about though with phospholipids is that some can be modified chemically to act as signaling molecules, right? And this is really interesting. These move around in the phospholipid bilayer, activating other molecules like enzymes. And so alternatively, they can be hydrolyzed to release little water-soluble glycerol-related molecules, which would diffuse through the cytoplasm and bind to specific receptors. So it's cool. It's like a messaging system that to where cells can chemically talk to itself, right, to its organelles, or to other cells. And so an example of one of these systems results in the release of calcium ions from storage that are being stored in ER. And it brings about exocytosis, which remember is like death of the digestive enzymes from pancreatic cancer cells, pancreatic cells as described in your book. Okay, so again, let me say that again. An example of one of these phospholipids acting as a way to communicate with the rest of the cell is where it enables the release of calcium ions from storage in the ER, which in turn brings about exocytosis of digestive enzymes from pancreatic cells. Okay, so cholesterol is a relatively small molecule. Cholesterol within the cell is super interesting. So cholesterol is found floating throughout the membrane, and again, it's relatively small. So remember, though, that that not only is there a lot of cholesterol, like in food, like a cheeseburger, but there's also a good amount that is in our cells. Okay, here it does a lot of good, a lot more good than clogging our arteries. Like phospholipids, cholesterol is a hydrophobic molecule. And so where do you think it wants to spend its time, right? Like, because alike molecules typically hang out with each other. Hydrophobic cholesterol is also located in the hydrophobic region of the cell membrane, right? That's created by the phospholipid tails. Okay, so cholesterol resides within the hydrophobic space and helps maintain membrane fluidity. It both provides structure and keeps the membrane from freezing. In addition, cholesterol helps the membrane act as a protective barrier to unwanted molecules. Okay, and you can see in this model that the cholesterol fits right in between those phospholipid tails. Okay, so and really in animal cells, the amount of cholesterol is the amount of phospholipids in the cell membrane. Plant cells have little to no cholesterol, and prokaryotic cells have none. And so, like I just mentioned, at low temperatures, cholesterol increases the fluidity of the membrane, which is a really good function because it prevents it from getting too rigid when it's cold. And this is because it prevents close packing of the phospholipid tails, right? So as the energy level decreases and, as kinetic theory would state, that those tails stop moving as much, they can't squeeze in real close because there are cholesterol molecules in there. So the increased fluidity during this colder temperature means that cells can survive during colder times. And it also helps to stabilize cells at higher temperatures when the membrane could otherwise become too fluid. Okay, so hydrophobic regions prevent ions and polar molecules from passing through the membrane. This is really important. One example is in the myelin sheath around nerve cells, because ion leaks would slow signals and thus affect our reactivity or cognition. And so again, this is an example directly from one of those little like pop outs from your book, one that you absolutely need to understand. Okay, so glycoproteins. So we talked a little bit about proteins, but we're going to talk more about glycolipids and glycoproteins. I mentioned them in the last lecture, but we didn't go into detail. So many of the lipid molecules on the outer surfaces of the cells are probably all of the protein molecules have short carbohydrate chains that are attached to them, right? And the carbohydrate chains project. They look like little antennae going into the watery fluid surrounding the cell, where they form hydrogen bonds with water molecules, and thus help to stabilize the membrane structure. So as you can look at this, or as you can see on this model, you can see the integral proteins, okay, and you can see the surface proteins, right? And then the ones that have the little antennae, okay, those are the little carbohydrate chains that are attached and bound, right, to the proteins. That's creating the glycoprotein. Okay. And again, as we talked about before, if you look, this is a really good model, so I really encourage you to pause this and really look at everything in here. You can see that alpha helix protein, okay, with the integral protein attached to the extrinsic proteins, right, which also have those little carbohydrate chains attached to it. Okay. So at low temperatures, oh, I went backwards, sorry. Okay, the carbohydrate chains form a sugary coating to the cell, and this is called glycolax. And in animal cells, the glycolax is formed mainly from glycoproteins, but in plant cells, it's really glycolipids. And proteins also play important roles in the membranes of organelles. Like in mitochondria and chloroplasts, they are involved in the process of respiration and photosynthesis. Okay, so carbohydrates. So we've talked a little bit about how those carbohydrate chains are creating the glycoproteins. Okay, not only are carbohydrates, food that we consume and bread and pasta and whatnot, they are also really important part of the cell membrane. The carbohydrate chains that are found on the outside of the cell membrane help to form those glycoproteins, and they can help with binding right, of other components that are coming into the cell, and they can also help cells adhere to other cells. So those chains, they allow the glycolipids to act as receptor molecules, right, and then bind with particular substances. And different cells, though, will have different receptors depending on the function of that cell. And there are really three major groups of receptor. There are signaling receptors, there's endocytosis receptors, and receptors that, again, aid in cell adhesion. So receptors, okay. Signaling receptors are part of a signaling system that coordinates the activities of the cell, right? And it's dependent on what activities each cell does. So a receptor will recognize a messenger molecule, like a hormone or a neurotransmitter, depending on what cell it is. And when the messenger molecule binds to the receptor, a series of chemical reactions will be triggered inside the cell, allowing that function to occur. So an example of this would be the signaling receptor that is in liver cells, which is waiting for the glucagon, right? So it's a glucagon receptor. When it comes in contact with glucagon, it signals for the liver to transfer glycogen into glucose. Okay. So a second group of receptors are involved in endocytosis. These bind to molecules that are parts of the structures to be engulfed by the cell surface membrane. Another group of receptors is involved in binding cells to other cells, otherwise known as cell adhesion, in tissues and organs of animals. Some glycolipids and glycoproteins act as cell markers, or antigens, allowing cell-to-cell recognition. So each type of cell has its own antigen. So it's kind of like a country with a different flag. So for example, the ABO blood group antigens are glycolipids and glycoproteins, which have small, tiny differences in their carbohydrate portions. Okay, and that's the ABO antigen specificity. So specificity. So the ABO antigens differ by just one sugar at the antigen terminus, and only the carbohydrate portion of the antigen is illustrated here. Okay. So many proteins act as transport proteins, and these provide hydrophilic channels or passageways for ions and polar molecules to pass through the membrane. So there really are two types of transport protein. There are channel proteins, which participate in facilitated diffusion. Okay, this is transport with no ATP or energy required, right? And then there are carrier proteins, which participate in active transport, and this is where ATP is required. So each transport protein is specific for that particular kind of ion or molecule. And so therefore the types of substances that enter or leave the cell are highly controlled. So channel proteins are water-filled pores, and they're gated. They allow charged substances, usually ions, to diffuse through the membrane. So this means that part of the protein molecule on the inside surface of the membrane can move to close or open the pore, kind of like a gate, which allows control of ion exchange. So two examples are the gated protein found in nerve cell surface membranes. One type allows entry of sodium ions, which happens during the production of an action potential. Okay, we'll talk about the action potential. Another allows the exit of potassium ions during the recovery phase, known as repolarization. So some channels occur in a single protein, while others are formed by several proteins combined. Okay, so pause the video, take a good look at this model. Carrier proteins can flip between two shapes, whereas channel proteins have that fixed shape, right? So the channel proteins are just participating in diffusion, so they don't have to change shape to allow anything to come in. It's very specific for their one thing. It's just a channel for diffusion. Where these carrier proteins, they have to be able to flip shapes. So as a result, the binding site is alternately open to one side of the membrane while the other is closed, and then switches so that the other is open. Okay, so if the molecules are diffusing across the membrane, then the direction of movement will normally depend on their relative concentration on either side of the membrane. Okay, they're going to move down a concentration gradient from a higher to a lower concentration. Okay, the rate diffusion takes place is affected by how many channel or carrier protein molecules there are in a membrane. Of course, you can only diffuse as quickly as there are places to diffuse through. And in the case of channel proteins, on whether they're open or not. So for example, in cystic fibrosis, it's caused by a defect in a channel protein in the cell surface membrane, including the ones that are, line the lungs. Okay, so this protein normally allows chloride ions to move out of the cells. And if the channel protein is not correctly positioned in the membrane, or if it does not open the chloride channel as it should, then the chloride ions cannot move out or cannot move out quickly enough. Okay, so other membrane proteins may be enzymes. For example, the digestive enzymes that are found in cell surface membranes of the cells lining our small intestines. So these will catalyze the hydrolysis of molecules like disaccharides. So some proteins on the inside of the cell surface membrane are attached to a system of protein filaments that are inside the cell, and this is part of the cytoskeleton. Okay, so these proteins really help to maintain and decide the shape of that cell, and they may also be involved in changes of shape when the cell moves. And proteins, of course, also play important roles in the membranes of organelles, which we know. So, for example, if the membrane of a mitochondria and the chloroplast, they're involved in the process of respiration as well as photosynthesis. Okay, so I want you to review and practice. So I want you to follow this link, and we can do this in class. You're going to play around with the construction of the cell membrane. This is a cool site. Okay. Okay, so we talked a little bit about cell signaling. I want to dive a little deeper into that. So, what is signaling? Basically, signaling is getting a message from one place to another. Okay, so take a second, look at this model. So, why do living organisms need cell signaling or need signaling? All cells and organisms have to be able to respond appropriately to their environments, right? So, this is made possible by a whole complex range of signaling pathways, which coordinate the activities of our cells. So, even if they are large distances apart, but they obviously have to be in the same body. So the basic idea of a signaling pathway can be summarized in a simple diagram. So you have a stimulus, right, or a signal like it being cold. You have a receptor, which picks up that stimulus. It transmits the message or the signal to the target or effector cell, which elicits a response. So you should have learned about this in STEM. Okay, so that signaling pathway includes receiving a stimulus or signal, transmitting the message, and making an appropriate response. A signaling pathway includes receiving a stimulus or a signal, transmitting the message, and making an appropriate response. Conversion of the original signal to a message that is then transmitted is called transduction. Okay, so I'm going to say that again. Conversion of the original signal, right, to a message that can be understood by the body is then transmitted. That process is called transduction. Okay, transmitting the message involves crossing barriers like cell surface membranes. And signaling molecules are usually very small for easy transport. Okay, so distances traveled may be short, as with diffusion within one cell, or long, as with long distance transport in blood or phloem. And usually, there are usually many components and different mechanisms along the route. So look at just this simple route for insulin receptor signaling. It is extremely complex. Okay, so signaling includes both electrical and chemical events and their interactions with each other. So, for example, the events that are associated with the nervous and hormonal systems in animals. These events involve a wide range of molecules produced by cells within the body, like hormones and neurotransmitters, as well as outside stimuli, like light, drugs, pheromones, odors, temperature, etc. Signaling molecules are extremely diverse, as you saw in that model. If they are hydrophobic, like the steroid hormones, like estrogen, they can diffuse directly across the cell surface membrane, and they'll bind right there to the receptors in the cytoplasm or the nucleus. So why does the cell surface membrane not provide a barrier to the entry of hydrophobic molecules into the cell? Like, why just let those in? Okay, I'd like you to study the boxes and the pop-outs in the book, and we will have a conversation about this in class. All right, thank you for watching, and we'll see